The relationship between protein misfolding rate and amyloid plaque formation. A meta-analysis of Alzheimer's disease by Group 5. Alzheimer's disease is a brain disorder where abnormal proteins flock together. A key protein called amyloid beta misfolds and then forms the sticky plaques that damages the brain cells. This study will examine how the speed of protein misfolding affects the plaque buildup in Alzheimer's disease. Our goal is to determine if faster protein misfolding is linked or connected to a more amyloid plaques. We also reviewed how the genes, environment, and the biochemical factors influences this process by looking at many studies. This study matters because amyloid plaques played a major role in Alzheimer's disease, because understanding what causes the proteins to misfold faster and could help us detect and treat Alzheimer's disease earlier and more effectively. This research will help nurses and healthcare workers to better understand how Alzheimer's disease develops. It could improve the patient's care strategies and the support to the development of new treatments. On scope and limitations, our study focuses only on the studies that measures protein misfolding and plaques in Alzheimer's, which excludes the other diseases. The data used were all previously published as we didn't collect new samples for this research. The research highlights that faster protein misfolding accelerates the amyloid beta aggregation into toxic fibrils and worsening neurodegeneration. For example, the generic factors like APOE3/4 increases the misfolding rates, elevating plaque burden and cognitive decline, while molecular chaperones mitigate this by enhancing the protein clearance, according to Sharma et al. 2025. Based on Golami 2023, Alzheimer's disease or AD causes damage to the central nervous system, which leads to the memory and cognitive impairments that is linked to neuron loss, neuron fibrillary tangles from hyperphosphorylated tau and amyloid beta plaques. Now, similar neurodegenerative diseases such as Parkinson's and Huntington's disease also involve harmful protein misfolding and accumulations, causing oxidative stress and neuronal damage, according to the study of Johnson et al. 2025. Now, failure in protein folding and degradation pathways is a key factor in AD, with gene expression patterns correlating with the cognitive scores. Based on the study of Young et al. 2023, advanced models like neural networks and support vector machines aids early diagnosis and treatment of design of AD by analyzing these molecular processes. In our general method, this study was conducted through meta-analysis, combining quantitative data from multiple peer-reviewed studies that focuses on protein misfolding and amyloid plaque formation in AD. A coding system was then used to extract the relevant data and ethical guidelines that were adhered to by using approved and anonymous secondary data. We applied statistical methods to investigate the relationship between misfolding rates and plaque formation. Graphs like forest plots summarize results, while checks ensures consistency and lack of bias. Our meta-analysis found a moderate positive link, R0.42, between faster protein misfolding and increased plaque formation. Heterogeneity was low at almost 21 and no publication bias was detected. Early onset Alzheimer's showed a stronger correlation, 0.55, between protein misfolding and plaques compared to the late onset cases, 0.29. This then suggests that biological susceptibility and aggregation kinetics vary by the age of onset. Post-mortem studies showed that the highest protein misfolding rates had approximately 7.4% more plate buildup than those with the lowest rates. This remained true even after adjusting for age and other factors. Based on the findings, the faster protein misfolding speeds up the plaque formation, which critically impacts the amyloid plaque formation, leading to the brain damage. This link is important by itself beyond just the total amount of amyloid present. Differences between the early and late Alzheimer's shows that early onset Alzheimer's shows the stronger relationship, likely due to the genetic factors and faster protein clumping, while late onset Alzheimer's is more affected by aging and reduced protein clearing capacity, like reduced proteosis and cellular stress. In conclusion, the increased protein misfolding rate is important in determining the amyloid plaque buildup in Alzheimer's disease. This deepens our understanding on AD and may guide future treatments. Now, on our recommendations, the study recommends improving the measurement methods of protein misfolding, like conducting longitudinal studies to clarify causality. Second, studying different amyloid beta types and genetic factors. Third, developing therapies to prevent the misfolding or enhance the protein clearance. And lastly, use misfolding rate as a marker for early detection and treatment planning. Thank you for your attention. I'm happy to present our Group 5's research paper.